Hi, all of our beloved students. How are you? How was the whole thing? Today I have prepared this video regarding cell biology from grade 11 textbook. Therefore, for those of the students who will be taking entrance examination this year, especially for those of the students taking entrance examination for this year. Therefore, if you have dropping the link of this video and if listening it somewhat better rather than accessing any other material, because the video is a short and a precise one. It includes starting from the definition of cell up to the end, that is called the major parts of a cell. The major parts of cell. In addition to this, the one is a short and a precise slides which support the wooden helps of a question. At the end of this slide, some questions are there that were mainly important for testing yourself, how to understand the slides, how the slides deal with, therefore how do you listening these slides. Therefore, after completing it, the final one, at the final, some few questions are there. The questions are mainly important for you if you're doing it critically. But before doing the question, clearly understand what I am said. In addition to this, regard the video after listening it and after doing the whole necessary things that was occurring in the one. If you have any comment, suggestion, it has been possible to share your ideas at the end, as well as if you have any question regard the prepared slides, if you don't understand, as well as regard the question, if any difficult question are there that have been possible to ask, as well as regard the theorem that have been possible to ask at the end of the video. At the end of the video, then all of you download. By downloading it, listen carefully. Okay, well, the main objective of this study is starting from the what is cell biology, because this chapter is including cell biology only, the starting from the history of cell biology up to the cell fractionation, including the major types of cell and the major parts of cell within their own function. After stating the history of cell biology, then we have been traveled to the what is cell theory and proposed by which type scientists, including size, structure, shapes of cell. In addition to this, assess the basic function of a cell. Afterward, all lives on Earth may originate from a cell. It may understand the one, and at the end, the types of cell, the major parts of cell, including the cell fractionation. Therefore, these main points are one of the major learning objectives which deal with that of cell biology. Okay, now we have been starting cell biology. Cell biology is one of the sub-disciplines of biology. Biology that study the basic units of life, including that of a cell. When you say the one, therefore, deal with all aspects of cell. The cell biology is general term. The cell which is included under that of a biology is deal with that of a cell biology. It deals with all aspects of the cell, including cell anatomy, cell division. Cell anatomy is one. Second one is cell division, which is including mitosis and meiosis. Mitosis and meiosis is one of the major parts of cell division. Meiosis including or occur in the normal body cell without including sex cell, that means somatic cell. But that of the meiosis, which is occur in the germ cell. Germ cell means including sex cell. For example, the human being is containing 46 chromosomes. Among this one, the 44 is known as called autosome. Normally, the 44 autosome is may give shapes for that of the born baby, that is called known as autosome. But that of the meiosis only including sex cell. Either the born baby is in order to classify as a male or other that of a male and that of a female. Therefore, male used for distinguishing a sex cell. That's called either male or that of female. As well, the cell biology including the process of cell, which such as cellular respiration and cell disease. When you say cell respiration, that cellular respiration process in the human being, the chemical reaction of cellular respiration is occur. The reverse of this one is called photosynthesis in plant cell. For example, in animal, by taking glucose plus oxygen, it has been possible to producing or yielding energy. The one is a major chemical reaction which is occurring that of cellular respiration. Apart from this, all living organisms are made up of cells. The one is the origin of a cell. We describe that one. As a result, cell biology is to deal with the all contents of life that was occurring on Earth, including a cell. And now that in biology, you know that the cell theory are there in biology. Cell theory is a scientific theory which is first formulated in the mid-19th century. That it says that all living organisms are made up of cells. As well as cells are the basic structural and functional units of life. In addition to this, all cells come from a pre-existing cell. That means without including spontaneous generation. That means spontaneous generation says that the cell is it comes from the non-living things. The one is a false idea. That is not true. Therefore, the one is proposed by that of the Lewis Pasteur. Lewis Pasteur says that the living things come from that of the non-living things, but the idea is not accepted nowadays because it is now proposed by that of scientific model. But the correct one that is accepted by the modern concept of cell theory is that a cell is come from pre-existing cell, which means from that of non-spontaneous generation. Yaw kana jechuun maal jechuudha? Boqonnaan kun waa'ee cell biology kanaas. Yaw hundi keessanii beektanii seeliin bu'uura lubbu qabeeyyii hundaa kan akka ta'e. Seeliin amma jirus seelii duraan jiru irraa akka dhufe. Seeliin kun maalirraa eegalee xumura orgaanizimii uumaa akka jedhus ni beektan. Seelii lama hoggaa wali dhufe maal kennaa tishuun qaamni sirni qaamaa dhumarratti maaluma orgaanizimii uumaa jechuudha. Kanaafuu isaa kan akka hubattan introduction dha waan ta'eef. ይሄ ማለት ነው ስለ ሴል ታውቃላችሁ ያው ሁላችሁም አሁን ያለው ሴል ድሮ ካለው ሴል ለማጣ ታውቃላችሁ ስለ ሴል ባዮሎጂ ትንሽ ካወራን ምን ይላል ሉዊስ ፓስተር እና ፍራንሲስኮ ሬዲን ከወሰድን በተለይ ሉዊስ ፓስተር ህይወት ያለው ነገር ህይወት ከሌለው ነገር ነው የመጣ ይላል ይሄ ግን ጥሩ አይደለም ምክንያቱም ጥሩም ሳይሆን ትክክል አይደለም ምክንያቱም ምሳሌ የሆነ ከብት ይታረዳል የታረደውን ስጋ በፌስታል አስራችሁ ልታስቀምጡት ትችላላችሁ ያ በፌስታል ታስሮ የተቀመጠው ስጋ አንዱ ለቀን ከተቀመጠ ትል ሊያመርት ይችላል ግን ስጋው ህይወት የለውም ትል ካመረተ ያ ስጋ ያ ትል ህይወት ይኖረዋል አይደለ ስለዚህ ሉዊስ ፓስተር ምን አለ ህይወት ከሌለው ነገር ህይወት ያለው ሊመርት ይችላል አለ ግን ሐሳቡ ትክክል ሊሆን ይችላል ይሄ ግን በሳይንቲፊክ ሞዴል ዲስከቨር አልተደረገም ምክንያቱም ህይወት የሌለው ነገር ከየት ነው የመጣው ህይወት ካለው ነገር ነው ምሳሌ ያ ከብት ካልታረደና ያ ስጋ ከዛ ከብት ላይ ካልተነጠለ ያ ስጋ ከየት መጣ ስለዚህ ግዴታ ህይወት ያለው ነገር ህይወት ካለው ነገር ነው መምጣት ያለበት እንጂ ህይወት ያለው ነገር ህይወት ከሌለው ሊመጣ አይችልም ስለዚህ ከሉዊስ ፓስተር ሐሳብ ይልቅ የፍራንሲስኮ ሬዲ ሐሳብ ነው ትክክል ሊሆን ይችላል እዚህ ጋር አንደርስታንድ ማድረግ አለባችሁ Yeah. Therefore, During the formation of this one, it made that the Louis Pasteur idea is says that a living things arised from the living one. The Francisco already says that a living thing is only arised from living things. Therefore, the one is the correct in the accepted idea. Yeah. A living things are only arised from the living one. The one is the best model which is accepted by that of the scientific theory. With the help of this one, with the advanced microscope today, now that two classification of compound microscope are there: scanning versus transmission. The one is it's able to identify or outlining the smallest structure of cell, which is not able to identify with that of the nuclei or that of the normal line. When compare these two types of the compounds electron microscope, the scanning electron microscopes is not a better as that of the transmission electron microscope. Transmission electron microscopes mainly used to identify a fine detail of an object, either unicellular or either multicellular, under that of the electron microscope. By the way, electron microscope is based upon electricity only. All of you know the one. At the results, when you compare transmission electron microscope versus that of scanning electron microscope, the transmission electron microscope have been better to identify the given structure of cell under that of the electron microscope rather than that of the scanning electron microscope. As keessatti kabeeku qabdan scanning electron microscope እና transmission electron microscope የኮምፓውንድ ኤሌክትሮን ማይክሮስኮፕ አይነቱ ናቸው። ግን ከእነኚህ ሁለቱም ኤሌክትሮን ኦር ኤሌክትሪሲቲ ባለው ቦታ ነው የሚሰሩት። ግን ትራንስሚሽን ኤሌክትሮን ማይክሮስኮፕ ከዚህ ከስካኒንግ በላይ የተሰጠውን ስትራክቸር በትክክል አርግቶ አጉልቶ ሊያሳይን ይችላል። ስለዚህ ይሄኛው በጣም ጠቃሚ ነው ማለት ነው። Now if you see the major parts of cell that was now today accepted by that of the modern cell theory, which is including that all now living things are made up of one or more cell. That is true. This means, by the way, all now living things are made up of one or more cell. This says that if the two cell is joined together, it have been possible to producing another cell. That is called a tissue. If the tissues are joined together, it have been possible to producing organ. Organ is going to organ system. Organ system is going to organism. If you take example, a human male and that of a human female. As we have been take an example from human male, if a sperm plus egg from that of the female. A sperm is single cell, egg is a single cell. If a sperm join with that of the egg cell, it have been possible to producing a zygote. A zygote is a tissue, but a sperm plus egg cell is a cell. That is called a single cell. When these two cells have been joined together, it have been possible to producing diploid cell. That diploid cell are called a zygote. Zygote is developed into that of embryo. Embryo is into that of the placenta. Through the process of this one, the final organism is developed. Therefore, all now living things are made up of one or more cells through the process of this one. Egg and the sperm cell is one of the best example that were describing the first modern cell theory that were accepted today now. The second one says that all living cells arise from pre-existing cell by division. The one is from that of non-spontaneous generation without including spontaneous generation. All living cells come from pre-existing cell by cell division. This means says that a living cell is come from a living cell. A living cell is not come from the non-living part. Therefore, during the process of this second idea, the Louis Pasteur methodology is not accepted because Louis Pasteur says that living things are made up of a non-living one. The one is not true, but this statement deals with that of Francis Corredi. Francis Corredi says that a cell is come from non-spontaneous generation without including non-living things. Therefore, today's existing cell, all living cells come from pre-existing cell by division. The third one, cell is a fundamental unit of structure and function of living organism. That means cell is basic structure and that of the functional unit of life. Ibeetani kana jechuun seeliin bu'uura lubbu qabeeyyii hundaati. Isa kana jechuun immoo seeliin amma jiru seeliin duraan jiru irraa dhufe. Seeliin kamiyyuu seel lubbu qabu irraa dhufe. Seel lubbu qabne irraa hin dhufne. Isa walii kana jechuun immoo organizimoonni hundi kamiyyuu seel irraa akka uumaman kan ibsudha inni tokkoffaan. Organizimoonni hundi seel irraa akka dhufan. እዚህ ምድር ላይ ያለ ኦርጋኒዝም ከሴል እንደመጡ ነው የሚናገረው። ምሳሌ ሰውን ሰዱ፣ የወንድ ስፐርም ሰዱ፣ የሴት ኤግ ሰዱ፣ እንቁላል ማለት ነው። ስፐርም ሲንግል ሴል ነው፣ ኤግ ሲንግል ሴል ነው። ሁለቱ ሲደመር ዛይጎት ይሰጣል። ዛይጎት ምንድነው? ደብል ሴል ነው። ዛይጎት እንደ ቲሹ ተቀበሉት። ስፐርምና ኤግ ሴል ጆይንት ታረጉ ሴል ነው፣ ቲሹ ይሰጣል። ቲሹን ዛይጎት አርጉት። ዛይጎት ፕሮዲዩስ ወደ ኢምብሪዮ ይሄዳል፣ ወደ ፕላሴንታ ይሄዳል። እንደዛ ኦርጋን ሲስተም ብሎ መጨረሻ ላይ ኦርጋኒዝም ህይወት ያለው ነገር ይፈጠራል ማለት ነው። ስለዚህ በዛ ፕሮሰስ ነው መሄድ ያለባችሁ። third one, activity of organism depend on the total activity of independent cell. That one is also true. The fifth one, energy flow, including metabolism in the biochemistry occur within cell. Metabolism is mainly responsible for the degradation and the building up of cell, which is including catabolism and that of the anabolism. The sixth one, cell containing DNA, which is found especially in the chromosome and the RNA found in the cell nucleus in cytoplasm. Also, this idea is linked with that of the first one. The final one, all cells are basically the same chemical composition organism of similar species. As the result, this seven main point is the major events which is accepted by the modern cell theory nowadays. Kanaafuu isaan torban kun yeroo ammaa kana keessatti yaa fudhatama kan argatanidha. Kanaafuu isa kana akka sirrii qabatan. The other one is how to discovering the cell starting from the ancient to the recent one. Inni amma lalu kun yaw akkaataa seelitti uumame jalqabarraa kaasee hamma ammaatti jechuudha. ይሄ ማለት የሴል አፈጣጠር ከመጀመሪያ ጀምሮ እስካሁን መጀመሪያ ምን ተፈጠረ ከዛንስ ምን ቀጠለ የሚለውን እናያለን። Therefore, the following events are the study of cell biology which represents the correct order from the ancient to the recent one. Ancient ድሮ ድራይገሊ hamma ammaatti jechuudha. ከድሮ ጀምሮ እስካሁን በመጀመሪያ ጊዜው the Carrier Jensen build compound microscope cell ከመታየቱ በፊት compound microscope ተፈጠረ። the first one. After the compound microscope is invented, Francis Corredi postulated living things. The living things do not arise from spontaneous generation. Francis Corredi maal jedhi lubbu qabeeyyiin hundi lubbu qabeeyyii irraa akka uumaman, lubbu maleeyyii irraa akka hin uumamne gabaase. Fourth one, Hawke. You know Robert Hawke described dire cell in a cork. Dire cell. As well as the fourth one, Leeuwenhoek view protozoa. By the way, the Leeuwenhoek only view protozoa, not identify the morphological and the basic the functional structure of protozoa. Only view protozoa. After viewing protozoa as well as may view bacteria. Then finally Robert Brown identified the nucleus as an important cell component because nucleus is one of the double membrane bound organelles which is found in both plant and that of animal cell. By including DNA mainly responsible for producing energy in plant and that of in both animal cell. Therefore, the one is the major important components of both animal and plant cell. Afterward, Schleiden that of Schwann introduced or proposed cell theory, and Dan Collier described mitochondria. Mitochondria is one of the poorest of cell, which is responsible for the production of energy in both animal and that of a plant cell. Then finally, Misker isolated DNA for the first time. DNA is, you know that, a double one, not single as that of RNA, because DNA is a major part of nucleic acid. Therefore, these are major events which is discovered from the ancient to the recent one. Afterward, Fleming described chromosome behavior during mitosis, because mitosis is one of the major parts of cell division, which is occurring in the normal body cell without including sex cell. Therefore, a 44 autosome in the human body cell is occurring that of mitosis. Afterward, Koch identified bacteria. Firstly, the Leeuwenhoek view bacteria with the help of a microscope. The microscope is called simple. Then Koch finally identified the morphological structure of bacteria. Then Golgi discovered Golgi apparatus. Rosca built the first transmission microscope. This transmission microscope is one of the major types of compound X-ray microscope, which is able to identify a fine detail of an object rather than that of the scanning electron microscope. Then Watts and Crick's proposed structure of DNA double helix. As well, after the first transmission electron microscope was invented, then laterally commercial electron microscope was produced. Afterward, on the recent one, the ship is cloned. Therefore, the major point that was in order to identify during D1 is especially for those of grade 12 students. Before going to explaining cloning of sheep, please all of you familiar with that of what is the main definition of clonings and that of genetic engineering የሚለው ማወቅ አለባችሁ። የክሎኒንግ እና የጄኔቲክ ኢንጂነሪንግ ልዩነት። Cloning means merely responsible for producing exact copies of genetically modified organisms. That's called cloning. But genetic engineering is merely responsible for transferring or a gene from one organism to the another one. By the way, that gene is taken from one organism by enzyme and inserted to another organism by using enzyme. And finally, it has been possible to producing genetically modified organism, but that genetically modified organism is not exact copies of the original one. That's called genetic engineering. But cloning is it has been possible to produce the exact copies of genetically modified organism. After the scanning electron microscope was invented, sheep was cloned. Afterwards, mice cloned. Sheep clone ከተደረገ በኋላ ከዛም በኋላ አይጥ ክሎን ከተደረገች then human genome DNA sequence draft completed. Therefore, neuron, cardiac muscle, blood cell created directly from reprogrammed blood cell. The one is the recent cell which is discovered. The recent cell. ስለዚህ ይሄ ቀደም ተከተል የሴል አፈጣጠር ነው። ከመጀመሪያ ጀምሮ እስከ መጀመሪያ ጀምሮ እስካሁን ያለውና ሁላችሁም ይሄን ስቴፕ ማወቅ አለባችሁ። ኢንተራንስ ላይ በደንብ ስለሚመጣ መጀመሪያ ምን ተፈጠረ ከዛን መጣ የሚለው ማወቅ አለባችሁ። አሁን ከያንዳንዱ እንጀምር። ከናፉ ኢኒሪፍኩን አካታ ኡመመሴሊ ጀልቀው ራካሲያን ጋማቲ ጀልቀው ማልቱ መሜ ማይክሮስኮፒ ወቴ ማይክሮስኮፒ ቢፋካሚቱ መሜ ኮምፓውንድ ዲሙ ኤሌክትሮን ዲሙ ላይት ዳሙ isa san erga adda baafannee booda akkaataa uumamasuulii tartiibaan ta'ee jira isa san qixa sirrii qabadha. Maaliif ni gaafatamtan waan ta'eef. Therefore now tokkoon laallaa jechuudha. Handi waan dinne jechuudha. Robert Koch is the first scientist. Robert Koch is one of the earliest scientists which is able to identify their cell network, not a living cell. Robert Koch jechuun by the way saayintiisa jalqabaati seelota microscope kallayidha. Microscope inni fayyadames microscope compound. Microscope compound kunis leensiin isaa yaa'u baay'ee qixa sirriitti magnification isaa guddisuu hin danda'u. Kana jechuun maal jechuudha? Robert Koch the first scientist which is able to identify dire cell in a cork, not a living cell, dire cell. The one is the Robert Hooke's compound microscope. The one is a cork. Under the cork, the hook drawing dire cell under that of a cork. After drawing dire cell in a cork, it may observe this dire cell under that of the compound microscope. Then finally, it have been concluded that the cell that are observing under that of the compound microscope with the help of 30 times magnification, the image is a very blurred. Is called a dire cell. Is that the conclusion of the Robert Hooke? Therefore, Robert Hooke only identified dire cell under the work. The magnification is not very good because only 30 times. As the result, it does not show fine detail of an object. Therefore, finally, a Hooke saw dire cells in that of a cork. Second one is Anton van Leeuwenhoek. Anton van Leeuwenhoek identified the living cell or the moving unicellular organism that is called protista and drops of water. Anton van Leeuwenhoek used simple microscope, no compound microscope. By using simple microscopes, it observed the moving unicellular organisms under that of a simple microscope. This moving unicellular organism is called animalcules, but that of an organism which have been taken from his teeth is called a tiny animalcules. Therefore, Anton van Leeuwenhoek identified animalcules. Animalcules is a cell which have been observed in drops of water that is called protista, but tiny animalcules is a bacteria which have been observed from that of his teeth. Therefore, the one is a discovery regarding that of the Robert Hooke. The other one is about that of compound microscope. Compound microscope is one of the main types of microscope, which is firstly invented by that of Zacharias Janssen. Therefore, as well, Robert Hooke have been using these types of microscope for identifying dire cell in corks. As well as the one is first microscope which is invented by the help of Zacharias Janssen. Therefore, the microscope have been using two objective lens eyepiece, including that of objective lens. Therefore, it have been possible to critically identify the given organism either unicellular or that of multicellular. And finally, it have been possible to critically identify the morphological structure of the given image under the help of this organism. Under the help of this microscope. The other one is Arindatrochetes. Arindatrochetes says that concludes all organisms are composed of a cell. Inni organism ከሚዩ ያው ሴል ይራከሉ ሙርቲሲ ሁሉም ኦርጋኒዝም ከሴል እንደመጡ ነው የተናገረው። ምክንያቱም እይታችንን ተጠቅሞ ማለት ነው። Therefore, this science says that all organisms are composed of a cell. This follow Manier's work in which he also discovery include stomata, osmosis, chlorophyll, including respiration. And finally says that all organisms are made up of cell. Therefore, the one is a discovery of that of the Arindatrochetes. Other one is Matthias Schlein Theodor Schwann. Matthias Schlein is a botanist. Theodor Schwann is a zoologist. Zoology who deal with that of animal cell. Botany which deal with that of a plant cell. Schlein and Schwann say clearly states cell theory. Cell theory at why cell theory worrying what I need. Therefore, Schlein and Schwann says that the cell is a unit of structural, physiological, and organizational living things. Therefore, one of the major proposed theory isolated with the help of Schlein and that of Schwann says that a cell retains dual existence. Cell is a basic structure and function of life, with including distinct entity and as well as cell is the building blocks of a life. But including this one, this up to this, these three are the accepted by that of the modern cell theory, but the one is not accepted because cell formed by free cell formation, which is called spontaneous generation. The one is not true. By the way, the cells come from pre-existing cell, that means from non-spontaneous generation. Therefore, from the discovery of shielding that of Schwann, this idea is not accepted. Therefore, the one is false idea, a false idea. Rather than this, the other part is a true. The final one is Rudolf Virchow. Rudolf Virchow, the most famous scientist, which is related directly or linked with that of the modern accepted cell theory, which say that omnicellular is cellular, which means all cell can only arise from another cell like it, by proposing the following one: all organisms are made up of one or more cells. It's true. All cells come from pre-existing cells. True. Cell is the basic structure and functionality of life. True. Also, cell retain dual existence is the synthesis and building blocks is the construction of organism. This also, all the four major main points are accepted by that of the modern cell theory. Therefore, Rudolf Virchow is one of the most famous scientists, which is directly linked or directly your work is directly deal with that of the modern accepted cell theory. That's called the work of Rudolf Virchow. Therefore, by summarizing the major events which is discovered by those scientists, these points are the measures accepted cell within the helps of modern cell theory. All of you understand, read the one critically. Okay, the other main important thing is to identify the how to measure the size of cell. In addition to this, what is the basic unit that was used for measuring the size of cell? The cell is measured by these units: millimeter, starting from meter. Meter is mainly responsible for measuring the largest cell, especially for that of the chicken egg cell. After meter, millimeter are there, micrometer are there, and nanometer are there. Therefore, meter is mainly responsible for measuring the largest cell, but nanometer is mainly responsible for measuring the smallest parts of cell. After having said the one, you know that cell size is directly linked with that of surface area to volume ratio. This means, in order to calculate the size of cell, the surface area to volume ratio is one of the measures important one to determine how the cell size may increase or that of it may decrease. As the result, when the cell gets bigger, the ratio of surface area to volume is decrease that. Look at this example. The one is the size of cell. May take size of cell starting from 1, 2, 3. Look at the size of cell is increased. The one is a cube, the one is the smallest cell, the one is larger than this, the one is also larger than this. At the size of cell increase, the surface area to volume ratio is decreased. Surface area to volume ratio of the size cell 1 is 6. Surface area to volume ratio of the size cell of 2 is 3. Ratio 1. The surface area to volume ratio of the sizes of 4 is 3 ratio 2, that is called 1.5. Therefore, as the size of cell gets bigger, surface area to volume ratio decreases. Look at 6, 3, size of a cell. A size of cell. Look at, for calculating surface area to volume ratio, understand the one. Height, width, length, how to calculate area of sides, how to calculate volume cube, how to calculate surface area, and finally how to include surface area to that of volume ratio. The area of one side is equal to this one. Therefore, we can write the one in the form of this. But when you summarize The main points which deal with that of surface area to volume as the cell size gets bigger, surface area to volume ratio is a decrease. Hammi cell hoggaa dabalaa deemu surface area to volume ratio akka xiqqaachaa deemu beekuu qabdan. Baddaatuuf volume to surface area is ይጨምራል የማወቅ አለባችሁ። የሴሉ ሳይዝ ሲጨምር surface area to volume ratio ይቀንሳል። ሪቨርስ ግን volume to surface area ይጨምራል። ምክንያቱም ማለት hammi ሴል hoggaa dabalaa deemu surface area irra volume irrata garuu garagaltuun isaa volume irra surface area ni dabala. Kana jechuun volume surface area caalaan dabala akkasumas surface area ni dabala volume ni dabala akkuma jechuudha. Therefore the one is how to calculate total surface area. One is a cube, 6 sides, the length and the width is 1. Therefore up to this, the one is total volume, the one is surface area to volume ratio. Look at our beloved students how to conclude regard surface area to volume ratio. At the cell size increase, surface area to volume ratio decrease. Look at the one. Look at the size of cell is 1. The size of cell in this is 5. But the surface area to volume ratio under the size of cell 1 is going to 6. But the surface area to volume ratio under the size of cell 5 is the final surface area to volume ratio is 1.2. Look at if the cell size is increased from 1 to 5, the surface area to volume ratio is decreased from 6 to 1.2. Therefore at the cell size increase, surface area to volume ratio decrease. But the reverse of this one, volume to that of surface area ratio is increased. As well, the volume is increased faster than that of surface area. That means the volume is increased as well the surface area is increased. Also, the one is the best example deal with in that one. Therefore, all of you see a home tech exam at may finish the one up to the final one and identify it. When the cell size increase from 0.5 up to 100, what about the surface area to volume ratio? Look at a small cell 0.5. The surface area to volume ratio is 12. The second cell size is Q. Surface area to volume ratio is 6. As the surface area to volume ratio decrease, as cell size is increase. Therefore, don't forget one. The final one is what is the importance of surface area to volume ratio? የሚለውን ገማል ፋይዳን surface area to volume ratio ማሊ cell size in UWS UTKT surface area to volume ratio. By the way, the major important points that is to deal with that of surface area to volume ratio is that the main role surface area is linked for controlling the amount of oxygen that was taken into the body cell, especially during the chemical reaction of cellular respiration. But volume is mainly responsible for controlling the amounts of energy released during the process of chemical reaction of cellular respiration. As a result, surface area to volume ratio is mainly responsible for releasing energy in the human body cell as well as in the other organism. Therefore, the one is the importance of surface area to volume ratio. Surface is mainly responsible for controlling the amount of oxygen taken into the body cell. The volume is responsible for releasing the amounts of energy which is broken down with the help of cellular respiration by taking glucose and oxygen molecule. That have been possible to releasing the large molecules of energy. Therefore, that energy is controlled with the help of volume. Therefore, the one is the importance of surface area to volume ratio. Mainly, the cell is responsible to release energy. The one is the importance of surface area to that of volume ratio. Also, the one is extra pointers which deal with that of surface area to volume ratio for more understanding and read carefully. Also the one is deal with that of that one. All of you test yourself. This is why it's important that cell have large surface area relative to its volume. Why does cell have large surface area relative to its volume? By the way, the one is a conceptual question. If you see what is the importance of large surface area relative to volume, the one is mainly used for respiring the cell in order to releasing energy. Therefore, all of you understand, remind this one. As well, calculate this and this one, and finally choose the best answer which deal with the correct. The other one is when the size of a cell increase, a surface area to volume ratio is what? Also the question. As the size of cell increase, surface area to volume ratio is decrease. The main point deal with this one. By the way, surface area of cell is mainly responsible for covering the membrane of cell, the outer part, which is mainly responsible for surrounding and enclosing the cell. But the volume is the amount of space occupied by the cytoplasm. The more explanation will deal with the equation is that as the cell become larger or bigger, surface area is increase more slowly than volume. This means volume is increased faster than that of surface area. Kan jechuun maal jechuudha? Hammi cell waggaa dabalaa deemu surface area akka volume dabalaa hin deemu jechuu barbaadeti baay'ee. Volume surface area caalaa akka malee dabala yoo hammi cell baay'ee dabalaa deemi. ማለት cell size እየጨመረ በሄደ ቁጥር surface area ይጨምራል። ግን surface area እንደ volume ሊጨምር አይችልም። ምክንያቱም surface area ወደ ውስጥ የሚገባውን ኦክሲጅን ነው ኮንትሮል የሚያደርገው። but volume የወሰነው ምግብ ላይ ተመርኩሶ ሪሊዝ የሚያደርገውን ኢነርጂ ስለሆነ ኮንትሮል የሚያደርገው volume ከsurface area በጣም እንደሚጨምር ነው ማወቅ ያለባችሁ። ግን የዚህ ተገላባጭ volume to surface area ratio በጣም እንደሚጨምር ማወቅ አለባችሁ። cell size ሲጨምር surface area to volume ratio ይቀንሳል። ግን volume to surface area ratio ይጨምራል ማለት ነው። ይጨምራል። ይሄም ማወቅ አለባችሁ። Also the one is a key point. Cell size is limited in accordance with ratio cell surface area to volume. Therefore, ይሄ main point ናቸው እንድታነቡት extra point ነው። Therefore, read it critically. The miss for the identification of that of. Okay, now we have been travel to the major parts of cell. Major parts of cell. That means types of cell. Look the one. Based on their originality, two types of cell are there: eukaryotic cell versus that of prokaryotic cell. Eukaryotic cell versus prokaryotic cell. Therefore, prokaryotic cell is the primitive cell which is evolved before that of eukaryotic cell. But some theories say that eukaryotic cells evolved before 2 billion years ago, but prokaryotic cells evolved before 1.5 billion years ago. But some theory say that when the primitive prokaryotic cells are set together, it has been possible to produce that of eukaryotic cell with the help of theory. That theory is called endosymbiont theory. Therefore, look, the one is the structure of eukaryotic cell, but the one is a structure of a prokaryotic cell. Look at the structure of cell which is found in all prokaryotic cell is known as plasma membrane, cytoplasma, ribosome, and DNA only. DNA, plasma membrane, cytoplasma, and ribosome are one of the cellular structure of cell which is found in all prokaryotic cell, including as well as in eukaryotic cell. But capsule, plasmid, pili, as well as fimbriae and flagella are a new structure of cell which is found in some bacterial cell or in some prokaryotic cell. Apart from This one, the other is found in all prokaryotic cell. Capsule, plasmid, pilla, and flagellum is found in some structure of bacterial cell or prokaryotic cell. Okay, therefore the one is the new cellular structure of cell, the new structure of cell which is found in prokaryotic cell. But the one is a structure of cell which is found in that of an eukaryotic cell. The one is in eukaryotic cell, eukaryotic cell. Look at. After identifying the structure of eukaryotic and the prokaryotic cell, it has been possible to classify the major types of this cell into three. The one is non-membrane bound organelles, the second one is single membrane bound organelles, the third one is double membrane bound organelles. Therefore, from this prokaryotic and eukaryotic cell, if you classify under those three organelles, look at chloroplast, mitochondria, and that of the nucleus. This is nucleus are known as a double membrane bound organelles. These are a double membrane bound organelles because containing DNA and are mainly responsible for producing energy. Therefore, nucleus, mitochondria, and that of chloroplast is known as called double membrane bound organelles. But the other one, vacuole, also endoplasmic reticulum, including Golgi body, as well as lysosome, the cell which is found only in that of animal cell. Lysosome are included under single membrane bound organelles. Vacuole, Golgi vesicles, or that's called Golgi body, endoplasmic reticulum, and the lysosome are found only in that of a single membrane bound organelles. Therefore, these are the basic structural features of eukaryotic cell, but the one is called a prokaryotic cell. Therefore, identify the similar. Okay, apart from after identifying the major parts or cellular structure found in both prokaryotic and eukaryotic cell, then we have been identified the difference between eukaryotic and that of prokaryotic cell, as well as similarity. That means comparison in the contrast between both prokaryotic and that of eukaryotic cell. Eukaryotic cell is containing a membrane bound organelles. Membrane bound organelles means eukaryotic cells have a membrane bound nucleus which is evolved from that of prokaryotic cell, but the prokaryotic cells do not containing membrane bound organelles such as a nucleus, while the prokaryotic cell do not including this one. As a result, prokaryotic cell is much smaller and simpler than that of eukaryotic cell. In terms of complexity, the prokaryotic cell is as well as less complex or simpler than that of eukaryotic cell because it not containing membrane bound nucleus. But eukaryotic cell is containing membrane bound nucleus. The main function of nucleus in eukaryotic cell is mainly responsible for storing genetic information. Genetic information is stored in the nucleus. For that of eukaryotic cell, while prokaryotic do not. As well, the difference in cellular structure of prokaryotic and eukaryotic cells, including the presence of mitochondria and that of chloroplast. Mitochondria and the chloroplast are not present in prokaryotic cell, but is present in that of eukaryotic cell. As well as the cell wall. For example, the cell wall is present in prokaryotic cell, but the cell wall of prokaryotic cell is made up of peptidoglycan. But eukaryotic cell is a difference. Plant cell wall, animal cell wall, and fungi cell wall, different cell wall are there. Therefore, each cell will have this difference, but the cell wall of prokaryotic cell is made up of peptidoglycan, including their structure of chromosomal DNA. Well, the other prokaryotic cell was the only form of life on Earth for millions of years until more complicated eukaryotic cell come into being through the process of evolution. Evolution. especially for those of grades 12 students. If you start the term evolution, it says that the all living things that was occurring on Earth is may come from the common ancestor. The one is a definition of evolution. The whole thing that exist in the Earth is come from a common ancestor. Therefore, prokaryote is the only form of life on Earth that existed for million years ago, before the eukaryote is originated. But all life on Earth now today consists of both eukaryotic cells. Prior to the origin of eukaryotic, all life on Earth was prokaryotic. Yeah, that means before the formation of eukaryotic cell, all life on Earth was prokaryotic. That is called lack of nucleus, or other membrane-bound organelles. In place of the nucleus, prokaryotic cell use nucleoids, which is bounded by including that of DNA called double helix. After the prokaryotic cell is formed, endosymbiotic theory states that a leading hypothesis called endosymbiotic theory says that eukaryotic arose as a result of fusion of archaeal cell with bacteria, where an ancient archaea engulfed an ancient aerobic bacterial cell. This means eukaryotic cell is originated or evolved when the primitive prokaryotic cell is associated together. It have been possible to produce eukaryotic cell. The one is the theory of endosymbiote. Therefore, the hypothesis is accepted, and I know that eukaryotic cell is evolved from symbiotic association of prokaryotic. Yeah, eukaryotic cell is today's eukaryotic cell is originated from a primitive association of a prokaryotic cell. You know that archaea, bacteria, eubacteria, including cyanobacteria, is a prokaryotic cell, but fungi, plantae, animalia, algae, and processes, including protists, called eukaryotic cell. Therefore, the theory of endosymbiosis base explain the eukaryotic cell says that eukaryotic cell is evolved from prokaryotic cell, engulfing another prokaryotic cell. Yeah, how the eukaryotic cell is originated from prokaryotic cell. This theory was proposed by that of endosymbiosis theory. The one is process how eukaryotic cell is evolved from the prokaryotic one. The steps of endosymbiosis theory. In the first step, the plasma membrane of ancestral prokaryotics have originated. This ancestral plasma membrane from that of prokaryote give rise to the endomembrane components, including nucleus, endoplasmic reticulum. Therefore, this is known as proto-eukaryotes. That means pro-eukaryotic, before the formation of eukaryotic cell. This nucleus give rise to the second. In the first endosymbiotic events, the ancestral eukaryotic consumed aerobic bacteria that evolved into mitochondria. The one is the second process in the formation of endoplasmic reticulum. On the third, a second, the one is by the way first endosymbiotic theory. On the third, a second endosymbiotic event in the early eukaryotic consumed photosynthetic bacteria that evolved into chloroplast. The photosynthetic bacteria are called cyanobacteria. Cyanobacteria are containing photosynthesis. Therefore, every plants have been possible to prepare their own food, which is classified under cyanobacteria. Therefore, cyanobacteria is classified under this third events. After the formation of aerobic bacterium, then the one is classified into photosynthetic bacteria versus mitochondria, or including modern heterotrophic eukaryotes. Heterotrophic means for those of the organism which is unable to prepare their own food. By depending on the host, it has been possible to produce their own foods. And finally, modern photosynthetic eukaryotes is produced. Therefore, the one is the basic steps how the eukaryotic cell is associated from the primitive prokaryotic cell. It's called endosymbiosis theory. But the one is evidence for the endosymbiosis theory. Evidence jechuun endosymbiosis theory akka maal jedhee yaa'u prokaryotic cellin jalqaba uumamee isaan sun walitti dhufanii eukaryotic cell uumanii jechuu isaati. Yaa'u prokaryotic cell kan dhugulasi dhugaa of keessaa hin qabne that's called prokaryotic cell. Therefore inni kun ይሄ ማለት መተማመኛ ኢንዱሲንግ ቲዮሪ እንዴት ኢዩካሪዮት ሴል ከፕሮካሪዮት ሴል ለተፈጠረ 5 ኤቪደንሶች አቅርበዋል። እነኛ 5 ኤቪደንሶች ናቸው። 5ቱም ኤቪደንሶች ስለማየት ማይቶኮንድሪያ እና ክሎሮፕላስት ይዛሉ። These evidences link with data including mitochondria chloroplast. Yeah, mitochondria and data chloroplast. Therefore, all of you read this one. Read this one. Therefore, origin of life, bacteria. Look at bacteria. Archaea, eukarya, eukarya, eukarya. From this diagram, what did you understand, our student? Prokaryotic cell is originated firstly before the formation of eukarya. Look at, archaea. Archaea is one of the best examples of prokaryotic cell. Therefore, archaea is closely common ancestor within that of bacteria rather than eukarya. Bacteria is deal with that of archaea, or archaea is dealing with that of bacteria. Then bacteria is closely related common ancestor within that of eukaryotics. Firstly, archaea is formed, then bacteria, then eukarya. Therefore, this diagram it may represent the origin of life, how the life on earth is originated from cell. Therefore, it may understand the one. As well, from this diagram, it has been possible to understand which one is related common ancestor within each other. Bacteria or archaea within bacteria, bacteria within that of eukarya. The same to this one. The 3 major domains of life had evolved by 1.5 billion years ago, as well as biochemical similarity shows that eukaryotic share more recent common ancestor within archaea. This diagram represents eukaryotic share more recent common ancestor. Recent means not till this time. Archaea is directly linked with that of eukarya. But our organelles probably descended from bacteria by endosymbiosis. Organelles undergo descended from that of bacteria. The other one is difference between that of eukaryotic cell and the prokaryotic cell. You know that all of you, prokaryotic cell is one of the simpler and the smaller cell. Prokaryotic cell have large surface area to volume ratio, which giving them that of higher metabolic rate and higher growth rate, and consequently they have a shorter generation time compared to eukaryotic. This means prokaryotic cell have large surface area to volume ratio rather than that of eukaryotic cell. If the prokaryotic cell have large surface area to volume ratio, the cell size of prokaryotic cell is smaller. As the cell size gets smaller, surface area to volume ratio increase. When the cell size gets bigger, surface area to volume ratio decrease. And the result from this theorem, prokaryotic cells have large surface area to volume ratio. That means the cell size of prokaryotic cells gets smaller. The other one, in eukaryotes, mitochondria and chloroplast have various metabolic processes and are believed to have been derived from endosymbiotic bacteria. Well, in prokaryotes, similar processes occur across cell membrane. Endosymbionts are extremely rare. In prokaryotes, endosymbionts are rare because these endosymbiont theories are proposed for the formation of only eukaryotic cell. The cell of prokaryotes are generally formed of different molecules called peptidoglycan when compared with that of eukaryotic cell. As well, prokaryotes are usually much smaller, simpler than that of eukaryotic cell. The one is okay similarity between that of prokaryotes and eukaryotic cell. Both prokaryotic and eukaryotic all cell, whether prokaryotic or eukaryotic, share these 4 features. Ribosome also present in eukaryotic, but the number of ribosome in eukaryotic is larger than that of prokaryotic. Also, the plasma membrane is present in both eukaryotic and that of prokaryotic cell. Cytoplasma is also present in both prokaryotic and that of eukaryotic cell, and the one is ribosome is also present, including DNA. DNA is also present in prokaryotic, but the DNA present in prokaryotic cells circular. This is look at DNA circular in prokaryotic cell. DNA circular, but DNA in eukaryotic cell is linear. Look at DNA in eukaryotic cell is linear, no circular. One is the most common for both prokaryotic and that of eukaryotic. The one is a comparison between eukaryotic and prokaryotic cell. Nucleus in eukaryotic cell present but that of absent. Therefore, all of you, the one is a comparison between prokaryotic and eukaryotic cell. Number of chromosome, cell type means either multicellularity or unicellularity. The membrane bound nucleus, which one is the membrane bound nucleus? Example including, as well as genetic recombination, lysosome, microtubules, endoplasmic reticulum, including mitochondria. Therefore, these all organelles are mainly used for comparing between that of eukaryotic cell versus prokaryotic cell. And all of you identify this one critically. Up to this, including the cell size. Up to cell size. Look at the size of eukaryotic cell is larger than that of prokaryotic cell. As a result, the size of cell for that of eukaryotic is complex rather than that of prokaryotic cell. Look at number of ribosomes, larger in eukaryotics, smaller in prokaryotics. Chloroplast present only in plant in that of eukaryotic cell. Chloroplast absent, but in cyanobacteria is one of the major class of prokaryotic cell. As a result, cyanobacteria have a chloroplast because they have a photosynthesis in prokaryotic cell. Don't forget the one. The one is summary of both prokaryotic and eukaryotic cell. Summary of prokaryotic and eukaryotic cell. Mitochondria from the chloroplast evolved from prokaryotic organism in that one because evolutionary theory says that eukaryotic cells evolved from the primitive prokaryotic cell. Also, eukaryotic cell would go on to evolve into diversity of eukaryotic. We know that today. The one is summary of both prokaryotic and eukaryotic cell. Please, all of you, after identifying difference between prokaryotic and eukaryotic cell, test yourself, please, for 5 minutes. The first question says that when did the first eukaryotic cell evolve? That means evolutionary formation of eukaryotic cell. Second, describe endiosymbiont theory. What is endiosymbiont theory? Says that which type is identified. Third one, a DNA in prokaryotic cell is by the way, the one is in prokaryotic cell DNA is circular, not linear, but in eukaryotic cell DNA is linear. Therefore, DNA in prokaryotic cell is circular, that's not bound with that of a protein. Therefore, the answer is look at this one. No associated with that of a protein. Okay, the other one is major parts of cell and their function. Major parts of cell and their function. Cell membrane or the plasma membrane are one of the major parts of cell, which is a living layer at the cell surface that isolate cell from the environment. The major function of this cell membrane or the plasma membrane play a crucial role in the cell and that of in the environment. The first one is enclose organelles and the other contain cytoplasm, provide mechanical support just like the same cell wall to allow the exchange of substance enter and exit, which substances enter into the cell, which substances move out of the cell. Therefore, these systems are generally controlled with the help of plasma membrane. As well, when the substance move into and out of the cell within the process of a diffusion is controlled by that of the plasma membrane. The other one is cell signaling, mainly used for recognizing hormones and immune system in animals and plants, including a growth regulatory hormone. These are the basic function of the plasma membrane in the cell. The plasma membrane moves substance in and out of the cell by simple diffusion, fast diffusion, osmosis. These are in the process of passive diffusion without requiring energy. Active transport, endocytosis and exocytosis are mainly in the process of active transportation system. These are called active process which are requiring energy. But from this, osmosis are responsible for moving water potential from high to low. What is a plasma membrane like? What is a plasma membrane like? One is another topics. Plasma membrane is a phospholipid bilayer. You know that. Plasma membrane is phospholipid bilayer. Therefore, plasma membrane or cell surface membrane is the membrane at the surface of the cell that isolates cell from the environment. When the cell when substance move into the cells and move out of the cell, it is controlled by the plasma membrane, as well as the cell is isolated between the cells and the environment with the help of cell surface membrane or plasma membrane. When the exchange of materials occurred in the plasma membrane, it has been possible to allow or control the exchange of substance between the cell that of the environment. These all are broken down with the help of cell surface membrane or plasma membrane. The other one is key events in developing the current model of membrane structure, which represent a correct order from the ancient to the recent one. Isaan kun yaw akkaataa uumama membrane cellii jalqabarraa kaasee hanga ammaa eenyu akka eegale eenyutu jalqaba propose godhe ammoo ማለት ሴል ሜምብሬን አካሂዱ አፈጣጠሩ ነው የሚያሳየው። ስለዚህ በትክክል እንድትይዟት ፈልጋለሁ። ምክንያቱ የመጀመሪያ ምን ተባለ ማን ነው ስለዛ አይዲያ የተናገረው የሚለው ነው። Therefore the first one, lipid nature of the membrane. By the way, the one is proposed by Charles Overton. Before Charles Overton, Robert Hooke states the idea of cell. That cell is called dead cell in a cork, but there's no idea about that of cell membrane. But Charles Overton says that proposed lipid nature of the membrane. That means the lipid is also present in the cell membrane. The second, lipid monolayer. Lipid monolayer means the lipid is present in the cell membrane, but the lipid is not double, not bilayer. It is a mono, single. The one is a single, which is discovered by Langmuir. Third one, a lipid bilayer. After the lipid monolayer or single lipid bilayer in the cell membrane is discovered, double or the bilayer of lipid is proposed by Gorter and Grinder. Yeah. Plasma membrane is a phospholipid bilayer which is proposed by Gorter and Grinder. After this one, lipid bilayer plus protein. Lipid bilayer plus protein. From this up to this, only the lipid is present in the cell membrane. But Dawson Danieli model says that the protein is also present in the plasma membrane or cell membrane. That's called sandwich model. Says that Dawson Danieli model called sandwich model. Then on the fifth one, Robertson, yeah, built based on that of the electron microscope. It has been proposed evidence to support the work of Dawson-Danieli model. And Robertson suggests unimembrane model, and he proposes that all membranes are essentially the same. The one is the work of Robertson, unit membrane model. And finally, Singer-Nicholson identify fluid mosaic model. The now today's widely accepted model is called fluid mosaic model. Fluidity is deal with that of lipid, mosaic is deal with that of protein. And finally, membrane protein structure is proposed by Hanoi and Henderson. Also, the one is hierarchical order, starting from ancient to the recent one. Also, the one is the same to the previous one. Lipid nature of the membrane up to the fluid mosaic model. Singer and that of Nicholson. The same idea with that of the previous one. Okay, now I've been starting from Dawson-Danieli model. We have been seeing one by one. This Dawson-Danieli model, the fluid mosaic model, is one of the model of plasma membrane. Dawson-Danieli model says that plasma membrane possesses protein that of phospholipid. It termed as sandwich of protein in the phospholipid. As a protein form bread of sandwich with a phospholipid for filling. Therefore, this model is not supported by observational evidence, as well as this model have been no use scientific experiment. By the way, for the acceptance of one model, it has been possible to undergo prediction hypothesis by identifying their own problem. But the Dawson-Danieli model, only by looking the surface of the membrane, it has been guessed only without undergoing this prediction. Therefore, the one is the weakness of Dawson-Danieli model. If this model is experimentally tested and if the hypothesis are accepted, and finally the results may not be rejected. But this model is not tested or in terms of scientific experiments, as well as not hypothesized, the prediction is not going on. As the result, this model is not supported by observational evidence. Therefore, unable to understand exact arrangements of phospholipid in the protein membrane. This means the Dawson-Danelli model, it doesn't understand why the protein, look at why the proteins occur at the top of the phospholipid bilayers and why the proteins occur at the bottom of the phospholipid bilayer, including why the phospholipid bilayers occur at the middle. It doesn't identify this one critically because the one is not tested with the help of experiment. As the result, finally, is not supported. Not supported. As the result, the model is therefore to be rejected. In addition to this, Dawson-Danelli model, in other words, lipoprotein sandwich model. The second one is fluid mosaic model. The one is also called the models of plasma membrane, which is proposed by Singer and Nicholson. This model retains accept the idea of phospholipid bilayer but rejecting sandwich arrangements. Kana jechuun maali? Phospholipid bilayer fudhatee sandwich model kuffise jechuudha. Phospholipid bilayer ወሰደ accept አደረገ ግን sandwich arrangement ጣለው reject አደረገው ምክንያቱም ከሱ ጋር ሄደ። Therefore, this fluid mosaic model states as membrane is a mosaic of a protein, not a sandwich. Membrane is mosaic of protein, not a sandwich immersed in fluid bilayer of phospholipid. Therefore, Singer and Nicholson have been used mosaic part of protein in place of sandwich, but the protein is arranged in different points. They also suggest as the arrangement was not static. Not static means the arrangement was moving, but was fluid and constantly changing its portion. ይሄ ማለት ነው arrangement not static ማለት move ያደርጋል ማለት ነው change ያደርጋል. The arrangement is moving and change its position from place to place. That arrangement is called the mosaic of a protein. As well as the fluids and the constantly changing its position. The one is the widely today's accepted models. Look at one of the key features for the acceptance of fluid mosaic model, starting from that of integral protein. You know that integral protein. The key feature of the model, as we currently understand it, say that phospholipid bilayer is the base for the membrane. The one is the fluid mosaic model. Phospholipid bilayer is the base for that of the membrane. In addition, integral protein, also known as, look at from this main points, integral protein is the intrinsic protein. Look at the one. The one is integral protein. The integral protein is known as intrinsic protein and it's transmembrane protein. Look at intrinsic. Intrinsic means inner part, including transmembrane protein, which is called integral protein. Therefore, this protein expands the membrane. Some of this protein play important role in moving substance across the membrane. These integral proteins are mainly used for expanding or moving substance across that of the membrane. The main part of this integral protein that was used for the transporting of a protein used are channel protein. Channel protein is called this one. Channel protein means in the form of iron. The one is the structure of integral protein. Also this is integral protein. Yeah, these are called channel protein. Mainly used for moving iron-containing molecule across that of the membrane. The other one is called carrier protein. These carrier proteins are mainly responsible for moving large molecules or medium-sized molecule across that of the membrane. Yeah, mainly responsible for moving larger molecules in that of medium-sized molecule across the membrane. That is called carrier protein. The other one is called peripheral protein. But generally, integral proteins are mainly responsible for spanning both the phospholipid layer in the plasma membrane. That means the bilayer part, double bilayer of phospholipid have been moved across the plasma membrane with the help of integral protein. If you see about that of the peripheral protein, Peripheral proteins only spanning one of the two phospholipids. Peripheral protein mainly used for me moving one of the only span one of the two phospholipid layers across that of the membrane. That is called a peripheral protein. The peripheral protein have a lot of function by including across enzyme. In addition to this, others answer integral protein to that of the cytoplasm. For example, any ion containing molecule, large containing molecule, medium-sized molecule are the one is spun across the membrane. It may with the help of this one, because the one is mainly used for spanning only one of the two phospholipid bilayers. That's called peripheral protein. The other one is glycoproteins, or called glycolipids. Glycoprotein is a protein that was attached to sugar. Glycolipid is lipids that were attached to sugar. During the formation of this one, the glycoprotein, glycolipid act as a receptor sites for hormones and drugs, which is called glycoprotein. Those glycoproteins, in that of glycolipids, are mainly responsible for act as a receptor for inducing hormones, drugs, immune system that were produced in your body cell, mainly controlled with the help of glycoprotein and glycolipids, as well as carbohydrate component of each cell can be specific and allow identification of the cell by the immune system. These all are controlled by glycoprotein, that of glycolipid. Glycoprotein is not bilayer. Please be careful. Glycoprotein is not bilayer. Protein by itself is not bilayer. Only the lipid one is the bilayer, because including fluidity. Only move fluidity. Mosaic parties may undergo look like circle at the tops and the bottom of the cell membrane. The final one is cholesterol. Look at cholesterol. The cholesterol is mainly responsible for reducing the fluidity of the membrane across that of the cell membrane. The one is fluid. This fluidity is reduced with the help of cholesterol. Well, the polar heads of phospholipid molecules is called hydrophilic, which is containing the polar molecule, including waters, phosphate. At the bottom, non-polar tails of phospholipid molecule are there, which is called hydrophobic, which is including protein, which is hydrophobic part, protein, lipids, or organic molecule. Non-polar, which is soluble under only non-polar molecule. At the middle, bimolecular lipid layer containing the protein are there. Therefore, all of our students critically identify which one is the bilayer from protein, lipids. Know that, identify it please. The bilayer is the phospholipid one. The one is all about the fluid, the mosaic model, which is proposed to Singer and Nicholson. The other one is also, these all are the key feature of fluid mosaic model that was seen in diagram. Identified critically. That was seen in diagram. Look at reduce the fluidity of the membrane, cholesterol. It was in diagram. The final one. Why is the fluid mosaic model called a fluid mosaic? This is because mainly the fluid part is fluid. The fluid is mainly the reason phospholipid is the membrane that can move and change position easily. With the help of this one, Singer and Nicholson proposed this model. The nature of these fatty acids and the amount of cholesterol in the membrane both influence the fluidity of the membrane. That means the movements influence it. Therefore, the fluids may not move during the process of that one, because the one is due to the nature of fatty acid. Final one. The mosaic part of the name comes from away the protein in the membrane give patchwork appearance. Patchwork means look like circle at the top of the membrane and at the bottom of the membrane. Patchwork appearance ማለት ልክ እንደ ሰርክል እንደ ጨረቃ ከላይ ካለው ሴል ሜምብሬን እና ከስር ካለው ሴል ሜምብሬን ሰርክል መስሎ ይታያል ማለት ነው። ይሄ ምን ይባላል? mosaic part of the protein. Mosaic part of the protein, which at the middle and at the outside part. Well, finally, Dick, test yourself. What does it mean, the term selectively permeable of the plasma membrane? ማለት እንደሆነ identify. Selectively permeability of plasma membrane jechuun maali? ምንድነው selectively permeability of plasma membrane? ስለዚህ ለግማሽ ደቂቃ ራሳችሁን ጠይቁ፣ መልሱን መልሱ። ከከበዳችሁ መጠየቅ ትችላላችሁ። The other one, how do substance move across the plasma membrane? ሌላው ቀጣዩ substance የተለያዩ substances በፕላዝማ ሜምብሬን or cell membrane አክሚቲ እንዴት ሙቭ ያደርጋሉ? አክሚቲ ሙቭ ጉዳን otherwise not all particle can actually pass through the plasma membrane. This because of largely lipid nature of the membrane. to pass through the plasma membrane by simple diffusion, or every molecule to pass through the plasma membrane, it must be small, it must be lipid soluble, as well as non-charger. Non-charger means ion containing molecule or charger one, as well as sugar and amino acid. The one is a larger molecule. In addition to this, the non-lipid soluble molecule are not move across the membrane. The one is the nature of the movements of substance across the plasma membrane. If the ones organelles have been partially or actually fulfilling this characteristic feature, it have been possible to move across that of the plasma membrane. Based on the movements or substance across the plasma membrane, it have been possible to classify substance into two parts: passive process and that of active process. Passive process means the process that was only requiring kinetic energy without including no extra energy from the cell metabolism. In addition to this, the passive process moves substance from high concentration to that of low concentration due to concentration gradients. Generally, passive process do not require energy because movement starting from high to low. Active process, the one is require energy from the cell metabolism in the form of ATP to drive transport, because active process moves substance from low to high concentration against the concentration gradients. For the movements of substance from low to high, the energy must be required. That's called active process. Active process. If you see the main types of transportation system based on the presence or absence of energy, it have been possible to classify through passive process, active process. The major part of passive process including simple diffusion, facilitated diffusion, osmosis. But major parts of active transports including active process including active transport, endocytosis, exocytosis. These are requiring energy when move substance from low to high concentration. Therefore, if you starting from the initial one, simple diffusion is one of the major parts of passive process which moves substance from high to low concentration, especially mainly substance, fluids, liquids, gas containing the molecule particle can be freely move around this molecule from high to low concentration. By the way, when the particle move across this simple diffusion, there must be concentration difference between the two sides of the membrane. That means high part and that of the low part, or the concentration gradients to drive the process. As the diffusion proceeds, means as the diffusion increase, high concentration will decrease. As the diffusion will proceed, the high concentration will decrease and the low concentration will increase until the two concentrations are the same. At this point, there will be no further net diffusion. That is called simple diffusion. This means when the diffusion is proceeds, the high concentration of diffusion is decreased, but the low concentration of diffusion is increased until when the two concentrations come to the optimum. That means at equilibrium. Then finally, there is no further net diffusion are there. That's called simple diffusion. Therefore, if there was not formed, the rate at which diffusion across the plasma membrane takes place influenced by concentration gradient. That means bigger difference in concentration result in faster diffusion. If the big difference in concentration gradients are there under simple diffusion, the faster diffusion are existent than the smaller gradients. The other one is thickness of the membrane. Also the surface area of the membrane. Therefore, the rate of diffusion across that of simple process is influenced by concentration gradients, thickness of the membrane, and the surface area of the membrane. You know, all of you, surface area of the membrane. Surface is mainly responsible for controlling the amount of oxygen which is entered to the body cell. Volume is mainly responsible for controlling the amount of energy which is traveled from your body cell, or when the body cell taking oxygen and the glucose may release energy, including waters and carbon dioxide molecule. The second one is facilitated diffusion. If facilitated diffusion the same to that of simple diffusion, the particle move from high to low concentration gradients, but the ones require ion or carrier protein. That means child protein and the carrier protein are existed under facilitated diffusion. Child protein are mainly responsible for moving ion-containing molecule across different protein. The carrier proteins are mainly responsible for moving larger and medium-sized molecule depend upon the active transport. The media concentration is pumped. As a result, facilitated diffusion is require carrier protein and child protein. Therefore, facilitated diffusion is different from that of simple diffusion by requiring carrier protein that of ion protein. In addition to this, however, it differs that the particles must be helped to diffuse across the membrane, or the diffusion must be facilitated by carrier protein or channel protein with an ion. Therefore, the one is the difference between that of simple and facilitated diffusion. The same to simple diffusion, facilitated diffusion is influenced or affected by the same factor as that of simple diffusion, with the exception that there is no the actual surface area of the membrane that determines the rate, but the number of carrier protein and child protein must be present. The one is a difference, but the other one is also the same. The third one is osmosis. Osmosis is mainly responsible for moving water potential. The water molecule move from high to low, or in the help of osmosis. As a result, osmosis is selectively permeable membrane. Osmosis is selectively permeable membrane, because the one is mainly responsible for moving the water molecule from high water potential to the low water potential without requiring energy. When you look at during the movements of water potential from high to low concentration, the rate at which the osmosis proceeds is influenced by surface area of the membrane, the same to simple in the facilitated diffusion. Difference in water potential and distance in molecule must be influence the movements of water potential from high to low molecule. The one is all about passive process, the other one is active process. Active process, under active transportation system, particle move from low to high concentration. The one is against the concentration gradient. When the particle move from low to high, energy must be required. ATP is pumped, produced. The one is the process of active transport. The same to one endocytosis. Endo means inner part, the one, including the internal one. Endocytosis. In this process, endocytosis, large particles are engulfed by the cell. The large particles are engulfed by the cell. That means called by that of the plasma membrane. Therefore, for the movements of particles from high to low, energy must be required. The endo, inner part, but the exo is outer part. During the formation of this one, large particles are secreted. By the way, before travel to exocytosis, endo including phagocytosis, pinocytosis, and that of receptor-mediated endocytosis, both requiring energy for moving from high to low concentration gradients. The one is all about simple diffusion. We have seen previously the table show. The one is factor influence the rate of simple diffusion. Also, we have seen the facilitated diffusion for moving the molecule from high to low concentration. Carrier protein or channel proteins must be required. Also, the facilitated diffusion affected by the same factor that of simple diffusion, but not the actual surface area of the membrane that determine the rate, but the number of carrier protein or the channel protein must be existed. The one is factor which influences the rate of facilitated. Okay, the other main important point is deal with that of active and passive transportation system is for more explanation osmosis one. Kana hundi kisa wae kana misiri da gifanda sabab ni sas mi gafi kisa ni relati bifa interansi ba kisa to kuta problem hypertonic isotonic hypotonic ni dufan mali amu gafi na cerdo kan gafi ulfa da kana positif wae kana mi da gifanda በትክክል በተሞና አንዳንዱ ምክንያቱም እነኚህ ጥያቄዎች በጣም ይመጣሉ ግን አንደርስታንድ ብዙ ማተረጓቸው ስለዚህ እስቲ እስቲ አዳምጡ በጥሞና በትክክል then osmosis is one of the major process of active passive process, one of the major parts of passive process which move water potential from high to low across that of pressure permeable membrane. Therefore, the osmosis including 3 types of solution: hypertonic solution, isotonic solution, hypotonic solution. Hypertonic solution means they containing low water potential than the cell. Low water potential means more negative water potential than the cell is existed. In other words, when more water exists in the cell, that water when going to the outside part, that cell lose water and become shrink. That is called hypertonic. Isotonic means the amounts of water potential, that means having the same water potential at the same. In the inner part or in the outer part, the water potential the same, equal. That's called isotonic. Hypotonic means having high water potential than the cell. High water potential means less negative water potential than the cell is existed. That's called hypotonic. Therefore, if you see in animal cell, in that of implant cell, if this solution is existed, and finally what may going on? For example, what happened to red blood cell if the plastin is in different solution? Red blood cell is a human blood cell, or this is called animal cell. When this animal cell immersed in the hypotonic, isotonic and hypotonic solution, ስለዚህ አመኩን ሴሊ ቢሉታቲ ይኑዳዋን የሰው ሴል ነው። ስለዚህ ይሄን የሰው ሴል እዚህ ሶስቱ ስናደርግ ሪዛልቱን ነው hypotonic solution. Hypotonic solution is mainly responsible for gaining water. That means, look at the one is hypotonic. Inside the cell, low water are there. On the outside, more water are there. But the salt is the same. Therefore, when the hypotonic is moved from high water potential to low, the high water which existed in the outside cell is moving to the inner part. Therefore, hypotonic solution gaining more water and they become swell and burst in animal cell. That is called hemolysis. Fakkeenyaaf hypotonic ala bishaan baay'ee qaba keessa bishaan xinnaadha garuu saaltiin dasim bishaan guddaan ala jiru kun gara keessaa hoggaa deemee seeliin kun ni hidha adda bal'ata akkasumas gain ያደርጋል ማለት ነው። ውጪው ብዙ አለ ውስጥ ትንሽ ዋን ያለው የውጭ ውሃ ወደ ውስጥ ሲገባ ሴሉ ይወጠራል። ስለዚህ gain water ያደርጋልና burst swell. That's called hemolysis in animal cell. But hypertonic solution, look at inside the cell more water are present, the outside less water are present, but the salt is the same. Therefore, this move from, look at when the water is moved to the outward, the hypertonic solution lose water. When it lose water, look like the structure of cell in hypertonic solution. Therefore, this cell lose waters and it becomes shrink. Look, shrink. ውሃው ይሄዳል ሽሪንክ ያደርጋል ይበታተናል። ሃይፖቶኒክ ኒውት ውሃ ጊን ያደርጋል ይወጠራል በጣም ይወጠራል ይናፋል። that is called hypotonic in animal cell. In animal cell hypotonic gain waters and swell up become burst and co-hemolysis. Hypertonic lose waters and it become shrink. Look at shrink. But isotonic neither gain nor lose water, it stayed the same. The amount of water that was entered into the cell and the amount of water leaving into the cell is the same. It's equal, 50-50. What happened to onion cell if placed in different solution? Onion cell is a plant cell. Kanama cell is bikalotati. Onion cell is a plant cell. If is the difference between hypertonic, hypotonic and that of isotonic solution, look at. Onion cell is placed in hypertonic solution. The same to other cell. When the cell placed in hypertonic solution, previously I said that hypertonic solution inside cell more water are present, but the outside less water. Therefore, when the water move from the cell to the outward, the hypertonic cell solution loses water and it becomes shrink, and finally plasmolysis. That's called hypertonic in plant cell. If you see hypotonic in plant cell, inside the cell low water are there, outside more water are there, but the salt is the same. Therefore, when the water is moving to the inner parts of cell, the cells become gain water, and swell up and become turgid. Therefore, when the solution or the water molecule move into the cell in hypotonic solution, turgid pressure become increase. Because gain water. As a result, it's become turgid. ይወጠራል። ውሃ ወደ ውስጥ ስለገባ ይወጠራል። That's called turgid, a swell up in hypotonic cell. Therefore, water moving to the cell, increase turgid pressure, it becomes swell up because the cell gain water and finally become turgid. But in an isotonic solution, the movement of water in the cell and the movement of water out of the cell is the same, so no change, equal 50-50%. Therefore, these three types of solution as one of main parts of osmosis, which may distinguish from that of animal and that of in plant cell by taking red blood cell from animal and onion cell from that of plant cell. And all of you understand the one critically, please, all of you. Although the one is a factor which is mainly influencing the rate of osmosis, similar to that of simple diffusion, but difference are there. Osmosis may influence by water potential, as well as distance between the molecule, including surface area of the membrane. The other part is active transportation system. Previously, I have seen that one. Active transportation system is mainly deal with moving the molecule against the concentration gradients from low to high concentration. For the movements of this molecule, ATP must be required. ATP is required and it may be broken down to yield ADP plus organic phosphate. Therefore, for the movements of active transportation system, the molecular gradients, active transport, look the one. Low concentration of molecule in the outside of the cell are there. On the inner part or inside cell, high concentration of molecule are there. Therefore, for moving or spanning the low concentration of molecule to the high concentration, ATP is broken down to yield ADP plus organic phosphate. At the results, when the one molecule is moved from the low part to the highest part, the energy must be required. The one is called active transportation system. Look at endocytosis, also one of the major parts of active process which require energy for moving from low to high concentration gradients. Also endocytosis including phagocytosis, pinocytosis and receptor mediated endocytosis. For moving the molecule from low to high, the one is also require energy, Also these are with the help of energy it will be possible to move molecule from low to high concentration molecule. Look at endocytosis. Phagocytosis, it says that cell eating. When the molecule, this one, span or move from low concentration to high concentration, the energy must be required. Therefore, the one is within the help of decoration of pseudopodia or extension of plasma membrane to enclose large particle or even all organisms outside the cell. In addition to this, phagocytosis enclosed by pseudopodia. That means they form internal vesicle which is then moved further inside the cell. As a result, the phagocytosis known as cell eating. When this cell, a large cell which is enclosed by pseudopodia, moved or traveled to the inner part, it may be enclosed by pseudopodia, and that is called cell eating. The second one is pinocytosis, which is called cell drinking. They differ from phagocytosis in scale. The scale is very small, the one is larger. As a result, it involves ingestion of the smallest particle. Ingestion means degradation of large particle into the smaller particle. Therefore, the one is the ingestion of the smaller particle, but the particles are still too large to cross that of the plasma membrane. Look at by other method, and it does not require the formation of large pseudopodia to engulf the particle. Then finally, the one is require energy for moving molecule from low to high, as are the final known as called cell drinking. The third one is receptor mediated endocytosis. Also, these are one of the major types of endocytosis. During this one, the membrane is full to form vesicles only in a region where the particle have bound to the specific receptor. Look at the particle bounds to the specific receptor, bound to the specific receptor, and the binding is stimulate the unfolding. That is called the receptor mediated endocytosis. But endocytosis have been possible to move molecule from the outer part to the inner. Endo means in. Therefore, the molecule have been traveled from the outer parts of to the inner part. Look at from the outer part to the inner part. The molecules are entered. This one from the outer part to the inner part. That's called endocytosis. That is called endocytosis. Endocytosis. The one is a definition. Also, the one is a definition. The other one is exocytosis. Also, exocytosis is one of the major parts of active process for the formation of movements of a substance. The one is move substance from the inside to the outside of the cell, the reverse of endocytosis. Because exo means outer part. This process may require enzymes and the hormones are secreted. Again, ATP is required for moving a molecule from inside part of cell to the outside part. That one is called difference between exocytosis and that of endocytosis. Look at the one is passive transportation system. By division, molecule move from high to low. Fast division. By using current channel protein, molecule move from high to low. Concentration gradients. The one is active transportation system. With the help of ATP, molecule move from low concentration to high concentration. During the formation of that one, ATP is broken down to release ADP plus inorganic phosphate. That is the requirements of energy in active transportation system. The same to this one, active versus passive transportation system. Look, iron, oxygen, sodium, when move from high concentration to low concentration without the requirements of energy, diffusion is processed. Active, when the molecule move from high to low to high concentration, for example, take sodium ions, potassium. When the molecule move from low concentration to high concentration, ATP is broken down to release ATP plus inorganic phosphate. Therefore, the one is the main difference between active and the passive transport. If you see the major types of cell, from before entering into the classification of cell, all of you familiar with that of what is organelles. Organelles means an individual cell which may producing a specific function. By the way, these all cells are known as unspecialized cell or undifferentiated cell. Two types of cell are still specialized cell versus unspecialized cell. A specialized cell is like blood cell, nerve cell, reproductive cell. Unspecialized cell including like nucleus, mitochondria, ribosome, endoplasmic reticulum, etc. From the one organelles, individual cell which is mainly responsible for producing a specific function. From that one, nucleus is one. Nucleus is a double membrane bound organelles which occupy about 10% volume of the cell. Mainly, the nucleus are responsible for controlling the whole activity found in the cell. Based on the one, the nucleus is divided into three. Nuclear envelope, which is called double membrane bound organelles. The nucleus, which is called non-membranous part. Chromatin, which is called a double membrane bound organelles by including DNA, histone complex, which is associated to form chromosome. These are the major parts of nucleus. By the way, the nucleus, mitochondria, and the chloroplast are mainly responsible for producing energy because it's containing DNA. The other one is mitochondria. Mitochondria is one of a double membrane bound organelles. Therefore, to release energy by aerobic respiration that is stored in ATP. Two membrane are the mitochondria. Inner part crista and outer part is called matrix. The crista is mainly responsible for increasing the available surface area of the cell, but the fluid matrix is act at the sites or act at the initial one for releasing aerobic reaction to take place in the chemical reaction. Therefore, fluid matrix is where the reaction of aerobic respiration takes place in mitochondria. Therefore, act as a site for the initiation of aerobic respiration to take place in the chemical reaction. In addition, in other ways, mitochondria is known as called a powerhouse of cell because most of the energy that was stored in the mitochondria and released in the formation of aerobic respiration, especially in the human body cell, is within the help of the mitochondria. The other one is ribosome. Ribosome is also the one is not double single membrane bound organelles mainly responsible for to synthesize protein. But They are also bound to the membrane system of the endoplasmic reticulum. The one is bound to the endoplasmic reticulum, forming rough endoplasmic reticulum. Rough endoplasmic reticulum have a ribosome on the surface. Then have been possible to synthesize protein. Each ribosome comprises two subunits that are made from RNA in the protein. Therefore, the two subunits which mainly used for the composition of ribosome are called RNA and that of protein. Don't forget the one, because RNA and protein are mainly used. Hi, our students, please make to do this question for a few minutes. That is called what difference between chromatin and the chromosome. Just make at home exams and do critically. The other parties of cell is called endoplasmic reticulum. Endoplasmic reticulum is major parts of cells which is found in both plant and animal cell. As a result, this cell is called single membrane bound organelles. Therefore, the function of endoplasmic reticulum is a network of a membrane system found in equal cell. Based on the function, two types of endoplasmic reticulum are the rough versus a smooth one. The rough endoplasmic reticulum has a ribosome on its surface. As a result, used to produce the transportation of a protein, but the smooth has no ribosome on its surface. For the reason of this one, the smooth is concerned to lipid synthesis rather than transportation of protein and the carbohydrate metabolism and detoxification. These two are the major parts of endoplasmic reticulum, which deal with transportation of protein, lipid synthesis. And the other one is Golgi body or Golgi apparatus, which is found in single membrane bound organelles, especially both plant and animal cell. The major function of Golgi body or Golgi apparatus is used to modify and distribute protein to the appropriate parts of cell. Many of the modification in the Golgi body, especially protein, may be converted into glycoprotein. For example, many of the modifications are in the Golgi apparatus act as a kind of tag which determine the final destination of the molecule. Is very important to identify the movements of a protein molecule or transportation of protein across that of the membrane. Apart from this, the other cell is called lysosome, which is found only in animal cell. Lysosome in the centroid is the only one kind of cell which is found only in animal cell. Also, this cell is single, not a double membrane bound organelles. Therefore, the lysosome have no specialized internal structure and are surrounded by single membrane bound organelles. They are formed in the Golgi apparatus and contain digestive enzymes that break down cellular waste debris. As a result, the major function of lysosome is it containing hydrolytic enzyme. As a result, they can digest worn out or damaged the organelles of a cell. Also, lysosomes are particularly abundant in phagocytic white blood cell. Phagocytic means mainly responsible for cell eating, including white blood cell. Here is the from the lysosome digestive organ cell that have been engulfed. These all are the major function of lysosome. The other types of cell are the cell which is found only in plant cell. That's called chloroplast vacuole in the cell wall. But the cell is not mentioned here. The chloroplast vacuole, the cell wall, is only kinds of cell found in plant cell, but vacuole is found both in plant and animal cell. Rather than this, the size of vacuole or the vacuole found in plant cell is larger than that of animal cell. Therefore, vacuole is present in both plant and animal cell, but in plant, the vacuole is larger than that of animal. The first cell which is found in plants is called chloroplast. Chloroplast is known as sites of photosynthesis. In addition to this chloroplast is double membrane bound organelles, mainly responsible for the productions of energy. By the way, plant is able to prepare their own food by absorbing energy or ATP. By absorbing ATP, it's produced glucose or food containing molecule. Also, DNA is important. As well as they use a chlorophyll act as a pigment based on the two two sites are there, that's called grana and stroma. Grana is light dependent reaction occur in the presence of lights only. In the presence of lights, as well as grana is membranous region in the chloroplast. Membranous region mainly used for splitting water containing molecule into hydrogen and oxygen. But the stroma is light independent reaction occur in the light independent reaction. That means in the absence of light, in the dark reaction. As a result, carbon dioxide is included in the stroma. Also, thylakoid are there. Thylakoid is a flattened sacs inside chloroplast where photosynthesis takes place. Therefore, thylakoid is linked with that of grana. As a result, chloroplast is double membrane bound organelles by using grana and stroma. Vacuole is also single membrane bound organelles which is found both plant and animal cell, but plant have larger vacuoles than animal. Therefore, the main function of vacuole is to store cell solution of minerals, salt, sugars containing molecule in order to maintain turgidity of the cell, or used for increasing turgor pressures of the cell. Therefore, our waste material is stored in the form of minerals, in form of salt, in form of sugar, used for increasing turgor pressure of the cell in vacuole. The other parts of cell is called cell wall. You know that cell wall is the only kinds of cell found in both prokaryotes and eukaryotic cell, but the cell wall of prokaryote is made from peptidoglycan, but eukaryotic is unique, not the same to this one. Also, this cell wall in plant cell is mainly used to give the plant cell wall as strength and elasticity. Cell wall is for giving the plant cell to give a strength and that of elasticity. This one is all about the major parts of organelles or cell found in plants, animal, and also animal cell. The ends from the end parts of this cell biology is called cell fractionation, or how biologists have been able to study different organelles by doing cell fractionation. This cell fractionation technique is to separate the mixtures of organelles. That organelles is maybe single membrane bound organelles, maybe double membrane bound organelles, or that is non-membranous bound organelles. Those are separated based on their mass size as they vary. That's called cell fractionation. Therefore, larger mass, the smaller the mass of cell under centrifuge by spinning at low or high speeds, separated by cell fractionation techniques. If the mixture of organelles spun in centrifuge, the largest mass of organelles is settled out first at low speed, but the smallest organelles are settled out last at high speed. Kan jechuun maali? Centrifuge jechuun meeshaa dhangala'oof jajjaboo adda godhu. Centrifuge ማለት liquid እና solid part ለያይቶ የሚያወጣልን ማለት ነው። ስፐን እያሽከረከረ። ስለዚህ ትላልቅ ኦርጋነሶች ሴንትሪፉጅ ላይ ስፐን ሲደረጉ ቶሎ ሲተላውት ይሆናሉ በአጭር ጊዜ ውስጥ ክብደት ስላላቸው። ላርጅ ኦርጋንላንድ ስሞለስት ኦርጋነሶች ደግሞ ሲተላውት ላስት ነው የሚሞቱት፣ መጨረሻ ላይ ነው የሚሞቱት። በጣም ትልቅ ስፒድ ይኖራቸዋል ይፈልጋሉ፣ ምክንያቱም ሳይዛቸው ትንሽ ስለሆነ። ከነየቹም ማሊ ኦርጋነሶኒ ጉድጉዳ ሳፊሰት ቀረት ይደፈኒ ጀልቀበረት ይበው ማሊፍ ኡልፋት ሚሳኒ ጉዳማንቴ ገሩ ኦርጋነሎኒ ትጥቃው ከን ጫሳሉይ ሲል ትጥቃ ሳፊሰ ጉዳ በርባቱ that means ስፒዲ ጉዳ በርባቱ ከናፉ ኦርጋኒዝሞኒ ትጥቆን ኦርሴን ኦርጋነሎኒ yokan ጫሳሉይ ሲል ትጥቆን ኩን ጡሙራራት ይበው ጀቹላ therefore the large mass of organelles have been settled out first at low speed. That's called nucleus is the largest organelles, double membrane bound organelles, including mitochondria, including chloroplast. But the smallest organelles are settled out last at high speed, including ribosome, vacuoles, endoplasmic reticulum. The one is smallest mass of organelles. Also, the homogenized cell sample is stored in suspension for separating the basic structure of cell based on mass and size. It's identified with the help of buffer, isotonic and cold. Buffer means neutral pH, prevent damage to the structure of protein. Isotonic means prevent osmotic water gain or loss by the organelles. Cool means to reduce the overall activity of an enzyme. Therefore, this homogenized cell samples are mainly stored in suspension for separating large masses in the smallest mass at low speed in that of at high speed to settle out first in that of the last. Therefore, all of you or below the students, please critically identify what is cell fractionation and the main objective of cell fractionation. Yeah, the main purpose of cell fractionation is to separate the mixture of organelles based on size or mass. The large organelles are settled out first at low speed, but the smallest organelles are settled out last at high speed. The one is cell. Also, the one is equation. Cell. Model. From model. Also, the one is from model, but the one is delusions out of transportation system. Also, the one is deal with an osmosis. Look at as an example. We will have been seen the one. Suppose you took a container and divided into two halves by membrane, which is permeable to water, and level the valves as A and B. Finally, you add the hypertonic solution in A and the hypertonic solution in B. What will be the expected result of this experiment? This one is osmosis. Osmosis ni betani movement of water potential from high to low. Container ito kuni jira. Container alla. Yan container በሁለት divide አደረግነው በማ by membrane selectively by membrane አንዱን ኮንታይነር A አደረግነው አንዱን ኮንታይነር B አደረግነው ኮንታይነር ኢረቲ A ኮንታይነር ላይ ሃይፐርቶኒክ ሶሉሽን እንጨምራለን B ኮንታይነር ላይ ደግሞ ሃይፖቶኒክ ሶሉሽን እንጨምራለን ስለዚህ ሪዛልቱ ምን ይሆናል ነው የሚለው እዚህ ጋር ማወቅ ያለባችሁ ሃይፐርቶኒክ ምንድነው ሃይፖቶኒክ ምንድነው የሚለው ነው by the way in hypertonic solution inside the cell more water are present if the water is moved to the outer part the hypertonic solution lose waters and it becomes shallow but the hypotonic solution inside the cell low water are there but outside the cell more water are there if the water molecule move into the inner part of cell the hypotonic solution gain water and it becomes soil as a result burst after identifying the main definition there is an hypertonic and hypertonic solution it have been possible to answer the question also these are there within osmosis also this is osmosis the one is the end thank you regardless this prepared the slides up to now if you have any comment suggestion it have been possible to share your ideas on this channel and finally thank you so much for listening and watching the video ሐሳብ ካላችሁ ሐሳባችሁ ሼር ማድረግ ትችላላችሁ ካዳመጣችሁና ጥያቄውን ከሰራችሁ የከበዳችሁ ጥያቄ ካለ መጠየቅ ትችላላችሁ እንዳፈሩ አሁንም በዚህ ስላይድ ላይ ዝግጅት በዚህ ባዘጋጁት ስላይድ ላይ ድክመት ካለበት ጥንካሬም ካለበት ሐሳባችሁን ሼር ብታደርጉኝ ደስ ይለኛል ምክንያቱም ቀጣዩ ያው ተዘጋጅቶ አታች ሊደረግላችሁ ይችላል ስለዚህ ያው በዚህ ላይ እናንተን ሐሳብ ማወቅ ስለፈልግ ሐሳባችሁን ሼር አድርጉ ያደከሰን erga video kana buufatanii dhageeffatanii booda yaada keessan share na godhaa gaafis yoo qabaatan asuma irratti gaafachuu dandeessan slide keessas ta'e gaafii ani isin gaafadhe keessas ta'e gaafii isin ingiliis jiraachuu danda'a kanaafu channel kana irra yookaan mana barumsaatti yoo ta'e dhuftanii gaafachuu dandeessan waan dhageeffataniif galatooma amma sagganaaloo sila addaamatachuun thank you so much love you